If you need to know about the nervous system, you are in the right place. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And today we're going through a comprehensive overview of the nervous system, probably the most important system of the body you need to understand to be a good nurse, healthcare practitioner, or just a struggling student. So what we're going to do is go through every single topic you need to know about the nervous system, give you some practical examples as well as some test questions that your A&P professor may ask you on your next exam. So let's get right to it. So in the nervous system, there's three main goals: detect, process, and respond. Everything in the nervous system comes down to those three things. Now you think about it, detecting. We're detecting stimuli in our environment, both outside and internal environment. Then we have to make sense of it. We have to process that information and determine how to respond to that information. Now the whole goal of all of these, put them together, is to maintain our friendly concept, homeostasis. So basically a set point of internal conditions that we need to keep constant. The whole goal of the nervous system is to maintain that. So a couple examples of that is we have to obviously detect things first. Now again, detecting things, we can have either internal or external stimuli that we will be detecting. So as an example, I've got a spider on my skin right here. So this is the skin. And we have a spider that's touching our hair follicle. Well, underneath the hair follicle are actually these sensory receptors. They're going to be called mechanoreceptors, and it is connected to our friend, a neuron, right? So this is a neuron here. And a neuron is just the signaling cell of the nervous system. So in this case, the neuron's going to detect it with a dendrite. We'll talk about that later on. So this is the dendrite. Then it's going to send that signal that direction. It's going to get super excited. It's going to send it down something called an axon here, the end part of the neuron, and it's going to communicate with the central nervous system, which is going to be the brain and the spinal cord. Again, this is called the central nervous system. Now think of the central nervous system. Everything has to go through it in order to think about anything or respond to anything. So it's going to be Grand Central Station. So in this case, we've detected something outside of our body that could obviously harm us, and we're going to tell the central nervous system about it. And we'll get to what happens next in a second. But we can also detect things that are internal, right? In our inside environment, specifically in our bloodstream. So as an example, we have different receptors called chemoreceptors. I'll label that. It's a part of the dendrite of a neuron. And the chemoreceptors can detect, as you can probably guess, chemicals in the blood and send the information to the brain about that information. So maybe our blood glucose levels are low. Maybe you need to respond to that. Maybe our sodium levels are too low. We need to raise that up, etc. So different dendrites, right? Different receptors can detect different stimuli. But this whole section is what's called the sensory division of the peripheral nervous system. The sensory division is sometimes also known as the afferent division, which basically means every signal that we are sending is being sent towards the important structure, which will be the central nervous system. So afferent is just a fancy word that says towards the central nervous system. Okay, so that is all we have in terms of the sensory division of the peripheral nervous system. The whole goal is to detect things outside and inside of our environment, as well as in sending that information to the central nervous system. Now, that was obviously the detect aspect. Now, that being said, we also have the central nervous system that you can probably guess is going to process that information. When this signal reaches the central nervous system, we don't really know what it is. We have to send it to places for the neurons to kind of unpack what that stimulus actually means. Like, is this something that could harm us, or is it just like a brush of the wind going over our hair follicles, right? We need to understand that. Well, how do we do that? Well, in this case, we have to have what's called interneurons. And interneurons are going to be found in the central nervous system. I'll label him here. Here's an interneuron. Okay, and these guys are literally found between neurons, whereas this guy in the green, I didn't mention this, is called a sensory neuron. You could probably predict that. So sensory neuron communicates to an interneuron. But at this point, if we have something on our hair follicle, we need to tell somebody important about it. We need to obviously tell the brain about it, because this is going to be the main region where we're processing information. So this neuron's going to send an action potential, send a signal all the way up to the brain, specifically, I'll tell you right now, to a region called the thalamus, which will be the sorting center of the brain. We'll talk about that a little later on. And in the thalamus, there will be another neuron that will send it up to the cortex of the brain. The cortex, basically the outer part of the brain. And this is where the magic happens, y'all. The cortex of the outermost part of the cerebrum, the outermost portion of your brain, is going to have literally just tens of thousands of neurons in a very small area that are going to be processing this information. So I'm going to circle this right here and say this is where the processing or the conscious processing happens. Conscious processing. In other words, this is where you become aware of what is touching you. So when the signal reached here, you didn't really know what was happening. But as soon as that signal reached the cortex, that's when we understand what the heck is going on. We may look at that spider, we may be like, that's dangerous, we need to do something about it, right? But again, the central nervous system, the whole goal is to do what? It's to process that information. Now, in this case, maybe the chemoreceptors detect some sort of chemical that's wrong. Same thing will happen. We'll probably just process it in a different region, maybe like in the hypothalamus, which will be right here. But again, notice I'm drawing a lot of close together neurons. So you kind of see how there's some sections where we're sending a long signal with one neuron, and then other places we're basically communicating to thousands or tens of thousands of neurons in a small little section. What this would look like, interestingly enough, here as well as here, this would be called gray matter. I'm going to label that over here. This would be gray matter. What gray matter is basically tons of neuron cell bodies called the somas. Okay, so the soma is the cell body of the neuron. There's just like tons of them, really close together. I'm going to say they're close together, and they are going to be processing, okay? So anytime you see gray matter, there's just a bunch of neurons really close together, and they're processing, communicating back. It's basically, if you've watched football, you may see a football player's huddle where they all get really close together and they're communicating back and forth. That's what the gray matter is. Now, gray matter, fancy term for this, is when neurons are unmyelinated. There's no myelin here. Now, what the heck is that? That's a new term, myelin. Myelin is basically the insulation wrapping around long neurons. I'm going to say that again because that's important. Myelin is the insulation wrapping around long neurons, Now, look on this diagram here and show me where long neurons are located. Well, you may see, here's a long neuron, right? So you've got this long axon, you've got this long axon, you got this long axon, and even this one, long axon, right? Notice I'm saying long axons. That's a signaling branch of the neuron, and these guys here and so forth will all be myelinated. So I'm going to maybe draw that in a different color, maybe red. So these guys all would be myelinated. What does that look like, or what does that mean? Well, if these are all myelinated, right here, myelinated, that means there is insulation wrapping, and it's going to be long axons with insulation. Okay, the myelin is the insulation around the axons. And in this case, it's going to appear white. It's going to be called white matter. So anytime you see white matter, know that it's just a bunch of long signals, long axons traveling up and down and all around through them. Whereas anytime you see gray matter, that's where there's a bunch of neuron cell bodies close together, just processing. So a lot of the times you may look in a textbook and you see this diagram where you see like dark substance and then lighter substance in the brain or the spinal cord, right? This is the spinal cord, this is the brain. The gray is just a bunch of cell bodies. The white is a lot of long axons of neurons. Now, where does the processing occur? In the gray matter. Where do the signals get sent a long ways? The white matter. So I think really, and this is a good analogy, I think of the spinal cord as basically an elevator between the brain and the peripheral nervous system. So the sensory information comes in and it goes up to the brain. And we'll see in a second that motor information typically goes down and out. So think of this as just an elevator bringing up sensory information or down motor outputs to communicate to other cells. Very cool. So there's a lot of topics here that you're noticing. So what a teacher could ask you is say, hey, what would you find in white matter? What would you find in white matter? Well, you would say, well, you'd find myelin, first off, the insulation wrapping around long axons. So you wouldn't really see any cell bodies in the white matter. You would see just axons. Great. Whereas if you said, oh, what's in gray matter, you'd say, well, there's a lot of cell bodies. Somas, right, the main part of the cell body inside of the gray matter. And I realize it might help you to maybe draw this out. So like in the gray matter, what you would see is just like a ton of neurons, maybe called multipolar neurons, that are just really, really close together. There's really no clear axon, clear dendrite. They're all just really, really close together looking like this, okay? And that's how they can process, just like a huddle, okay? So again, what did we have? We had the sensory information coming into the central nervous system. Central nervous system kind of processes it out, sends the information where it needs to go to be processed. But now, probably the most important thing is we have to make some sort of response. So another aspect of the peripheral nervous system, so anywhere outside of the central nervous system, is going to be called the motor division. Okay, so we have the sensory division, the motor division, and the motor division is going to make an effect on different tissues. So it's going to make an effect, remember it's efferent, okay? The efferent pathway, where we're sending signals away from the central nervous system to contrast it to the afferent sensory, where we're sending information towards the central nervous system. So let's see what happens in the motor division. Well, your body has a lot of different tissues that need to be told what to do and when. The question is, when do they do things? Well, it's when the motor division of the peripheral nervous system tells them to do things. So as an example, this is your skeletal muscle. You have a lot of these skeletal muscles attached to your skeleton, and they are voluntary. So these skeletal muscles will be controlled by motor neurons. I'm going to erase a little bit just so I can get through here. Okay, so we've got some motor neurons that are going to be chilling in the central nervous system, interestingly enough, and we are going to have these guys come out and communicate to the skeletal muscle. Okay, so this is something called a lower motor neuron that can come and communicate directly to the skeletal muscle. Well, interestingly enough, you can, in a way, control him because you can control your skeletal muscles. Well, how does that work? Well, we know up in the cortex of the brain, there's another neuron called an upper motor neuron that can come down and communicate to him. So in a way, if you can control him, then you can control your skeletal muscles. Okay, so this is just the order of it, right? Upper motor neuron to lower motor neuron. Lower motor neuron communicates directly to the skeletal muscles. Again, sending the information away from central nervous system, it's the efferent pathway, and you can control this. So we call this specific section of the motor division the somatic motor division. Somatic motor division. And that is talking about voluntary neurons that contract or that stimulate skeletal muscle. Okay, skeletal muscle. So the somatic motor division consists of neurons that can contract or can stimulate your skeletal muscles. So that's really cool. You can consciously control this. Now let's relate this back to the analogy of the elevator, right? We're sending information down the elevator through white matter, out also through white matter, and getting to the skeletal muscles to trigger them to contract. Very cool. But you also have a variety of other structures in your body that you can't consciously control. Let's get some examples of that. This, for example, is your cardiac muscle. This is your heart muscle, right? You can't control that. It happens automatically. Here we have smooth muscle, things like around your intestines, maybe around your arteries. You can't control those either. And then lastly, you also have some structures like glands, maybe like your pancreas, maybe your stomach, maybe like sweat glands. And again, you can't control these consciously. So the question is, they need to know when to do their stuff. How do they get told to do it? Well, in this case, we need a new branch of the motor division, right? We're still affecting tissues, but we can't control this branch. So we call this new branch, I'm going to draw in a different color, the autonomic branch, the autonomic motor division. Now, the autonomic motor division, this is what's interesting. Your glands, for example, can do one of two things. They can produce a product and release it, right, into either like an exocrine gland where it's like a sweat gland going out of your body, or an endocrine gland where it's making a hormone into your bloodstream. But they can do one of two things. They can either secrete, or they can obviously not secrete, right? They can produce the product and put it in there, or they can not do it. Well, so we need to be able to tell them to get stimulated as well as to not be stimulated. Same thing with maybe your intestines. We need to tell them sometimes to contract and then sometimes to relax. Sometimes to contract, sometimes to relax. So you see how there's kind of this on and off switch for all of these different tissue types. Well, how do we tell them whether to turn on or off? We need two different divisions inside the autonomic division to tell them basically go or stop. So those two divisions are things called the sympathetic division as well as the parasympathetic division. I'll draw in orange. Okay, so sympathetic and parasympathetic. Now this is where it gets a little difficult, but really interesting. The sympathetic will prepare your body for something we call fight or flight. Fight or flight, basically amping you up in order to run away from something or to attack something. Whereas the parasympathetic is the opposite, and it's going to tell your body to rest and digest. Now, what do we mean by that? Well, the parasympathetic may stimulate certain things when you are digesting, but it will also inhibit certain things so that you can rest. Whereas the sympathetic may trigger some things, but also inhibit other things. So let's get an example here. I always like to think of these two as like, have you seen those old cartoons where you have like the angel on one shoulder and the demon on the other shoulder, and the angel's like, you should do this, you should be really kind to this person, and the devil's like, no, you shouldn't, you should attack them, and blah, 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 right? So you kind of have these two different voices exerting opposite effects. So what I want you to write with the autonomic division is these two divisions, sympathetic and parasympathetic, will basically talk to the same tissues, but exert opposite effects. Exert opposite effects. Very, very important. So let's take an example with smooth muscle. Let's say that we have smooth muscle, and let's say this smooth muscle is around your intestines. Okay, so we're looking at the intestinal smooth muscle. Intestinal smooth muscle. Well, let's think about it. We have parasympathetic neurons that will come in. I'm going to erase a little bit once again to just have some space. Okay, we're going to have parasympathetic neurons coming in and communicating to the smooth muscle. And we're also going to have sympathetic neurons coming in and talking to the smooth muscle. As just a quick note, parasympathetic neurons originate in the craniosacral regions. So the sacral nerves as well as the cranial nerves contain some parasympathetic nerve fibers. So we're going to say maybe this guy's coming in and talking to the intestinal smooth muscle. And then we're also going to have sympathetic neurons that are going to be originating in the thoracolumbar region. So basically the thoracic nerves to the lumbar nerves. And these guys are going to come in and communicate to the smooth muscle as well. Now, I'm making this a little simpler just to kind of make things easy for you. In reality, there would actually be two neurons involved in both. There would be a preganglionic and then a postganglionic neuron for both of these. So it'd look more like this. Okay, so we're not going to get that detailed quite yet, but just know parasympathetic and sympathetic will have kind of a two neuron system regardless. So let's think about this. When the parasympathetic neuron is firing, so we're in a state of, think of rest and digest. Anytime a parasympathetic neuron fires, we're going to prepare our body to rest and digest. What do you think it would do to the intestinal smooth muscle? Remember, we're in a state of digestion. So interestingly, the parasympathetic neuron will release something called, in this case, a stimulatory neurotransmitter. Neurotransmitter. That's basically the chemical that's being used to talk to the smooth muscle. And in this case, I said that it's going to be stimulatory. Why? We're stimulating digestion. We want our intestines to move during this point, right? But what if the sympathetic division is activated? Let's say we see a lion or we see a spider on our arm and we're terrified and we activate our sympathetic nervous system. Well, in that case, the sympathetic nervous system comes in, acts on the intestinal smooth muscle, and actually releases something called an inhibitory neurotransmitter. Inhibitory neurotransmitter. So in this case, it will actually basically relax the smooth muscle and tell it to just stop digesting in general because we're in a state of sympathetic fight or flight. So once again, remember my analogy. We've got like the angel and devil on our shoulder. We're communicating to the same structure, exerting opposite effects. Now, as an example, what about the heart muscle tissue? In this case, if you're in fight or flight, the sympathetic neurons would actually release a stimulatory neurotransmitter. So the sympathetic division stimulates cardiac muscle to contract more and harder and faster. Whereas the parasympathetic, when it releases a neurotransmitter to the cardiac muscle, would inhibit it. It would slow it down. It would relax its contractions because we're in a state of rest. So you see kind of the duality of the sympathetic and parasympathetic nervous systems. Depending on the tissue that we're talking to, it might exert the opposing effect. So as one more example, maybe you get this on a test. What if we talked about the salivary glands? What if these were, as an example, salivary glands? Think about it. In this case, the parasympathetic would likely, what? Stimulate them to produce saliva so that you can obviously digest your food better, whereas the sympathetic, if it was activated, would inhibit the salivary glands. So I can draw that here. So salivary glands, parasympathetic would stimulate, whereas sympathetic would inhibit. So you see, again, the duality of this. And this explains a couple of things, right? Let's say you're really anxious about a test and you kind of feel nauseous. Well, it's because your sympathetic neurons are activating and relaxing your smooth muscles, so you kind of feel that nausea going on. Whereas before, like races, I was an endurance athlete in college. Whenever I would be stressed out about a race, I would get dry mouth. I would get dry mouth. Why? I'm in a sympathetic state, fight or flight state, and it's talking to my salivary glands and inhibiting them so that I can't produce saliva. So therefore, my mouth is really dry right before a race, and it really annoyed me. Okay, so overall, this is a great subdivision of the nervous system. Sensory, afferent, sending signals towards central nervous system about some sort of sensory information, whether external or internal. And we've got to process it. We've got to send it to the right structures of the brain and spinal cord in order to understand what's going on. From there, we can respond by moving our skeletal muscles voluntarily. It's a somatic motor. Or we can use the autonomic division, sympathetic or parasympathetic, to affect different internal glands, smooth muscle, and so forth. Awesome. That's a great start. So now we need to talk a little bit more about neurons, right? And how they communicate back and forth with each other with action potentials and neurotransmitter types. All right, so now for the process that's most misunderstood by my students. So I have a really good way of teaching and understanding this. So now we're going to talk about action potentials and how neurons actually communicate to target cells. And no, the target cells don't actually look like that, but Target, if you want to sponsor me, feel free because my cell looks like your logo. And by the way, if this has been helpful so far, please consider liking the video, subscribing to the channel. Really appreciate your guys' support. So what do we start with? Well, we have a neuron here, and we're going to communicate to, again, a target cell. As we saw previously, this target cell could be another neuron as an example. So we could have a neuron talking to another neuron. We could also have it be talking to like a gland, like we saw earlier, or some sort of muscle tissue, okay? So typically, those are the things we can communicate with. But how do we communicate to that target cell? Well, we know the neurons at the end of their axon, so this is the signaling branch, the axon of the neuron. The axon will contain a little organelle inside called a vesicle. So I'm going to label that here. This is called specifically a synaptic vesicle. I'll tell you why in a second. And a vesicle, again, is just an organelle inside of a cell. So cells have tiny little organelles, and it contains a product inside. Now, this product inside is called a neurotransmitter. That's a fancy word for saying, hey, this is how neurons transmit or share information with somebody. Now, this neurotransmitter level of organization is a chemical. Okay, so this cell produced this chemical, packaged it into a vesicle, and if we release it into something called the synapse, the space between the neuron and the target cell, why it's called synaptic vesicle, we can then communicate to that target cell. So it's a chemical from neurons that relay a message to a target cell, Now, as we saw in the previous section, these neurotransmitters can be either one of two things. They can be stimulatory, or they can also be inhibitory. So we can either tell the cell to do something, or we can tell the cell to essentially stop doing something. It's just as important to turn something on as it is to turn it off, okay? Now the question is, how do we get this neurotransmitter out? And how does the target cell actually receive that information? Well, to the second point, that target cell always has to have a receptor for that neurotransmitter, okay? So I'm going to draw that this target cell must have a receptor of some sort, but we need to be able to get that neurotransmitter out. So how do we get the neurotransmitter out? We conduct something called, I'll write it up here, an action potential. Action potential. This is basically, as a definition, a flow of positive charges, if I can spell properly, down an axon. Down an axon. Now, what the heck does that mean? A flow of positive charges. Well, this is where there's sometimes some misconceptions. When we're looking at any cell, neurons included, the neuron always starts as a very, very negative charge. I'm going to just draw a bunch of negative charges around the membrane of the neuron. And the reason for this is because inside of any cell, we have a lot of a macromolecule called proteins. Cells produce proteins through protein synthesis, and these proteins usually have a net negative charge. So since there's just so many proteins packed inside the cell has in a way, negative voltage to it, which is super cool. So I want you to remember, when the cell is negative, it is doing nothing, okay? It is resting. That's why we call this voltage, around negative 70 millivolts, a resting potential, okay? Resting potential. It is negative, and it is doing essentially nothing. Okay, very important term. You'll probably have that on a test. Okay, so the question is, again, how do we release the neurotransmitter? Well, we need to have an action potential, a flow of positive charges down the axon. So essentially, we're trying to get this negative to positive. Just like you may have tried to take a pessimistic friend of yours and turn them into an optimist. Good luck with that. But we have better luck with neurons. So check this out. So again, end goal, make neuron positive. How do we do that? Well, the neuron has to receive some sort of stimulus, okay? So I'm going to draw this stimulus of some sort right here. And the stimulus is going to act on a specific region of the neuron that I drew here at the very end. This very end of the neuron, they kind of look like these little branched out sections of the neuron. These are called dendrites. And dendrites, always remember, dendrites detect. This is where we're actually going to be detecting stimuli, okay? Now, obviously, depending on the neuron, it might be a different stimulus. It might be a neurotransmitter talking to this neuron. It might be a spider crawling across your hair, and so we detect the mechanical force. It might be like at the back of your eye, it could be light, photons, right? So this could be a variety of different stimuli. So let me just list some of them. It could be, again, like a chemical, like a neurotransmitter. It could be a mechanical force, okay, something actually moving it. So pressure. It could be also maybe some sort of damaging stimulus, something that's actually harming your body. It could be light, okay? And I'm just going to say etc. Okay, a lot of different stimuli that these dendrites can detect, again, depending on what they are, okay? So we have a stimulus. What does that stimulus do? Well, the stimulus is going to open up some gates. This is a really weird concept if you haven't learned about cell biology. This is a cell. This is something called a membrane protein. This membrane protein can look a variety of different ways. You can have one looking like this, one looking like this, one looking like this. There are a variety of different types of membrane proteins that the cell produced and put itself into the membrane. Now, in this case, we're going to call this guy, as an example, a ligand-gated sodium channel. Okay, a few things to break down. A ligand-gated is basically saying this is a protein where if you have a key and it binds to the lock, the receptor, we are going to open that gate. Okay, there's also mechanically gated channels where if you put a mechanical force on it, it opens up. So as an example, we have the stimulus that's going to open this guy up. Let's just say it's like that spider crawling across your arm and it's going to be a mechanically gated or a ligand gated channel. So this guy will open up. This guy opens up because that little receptor right here was forced down. So actually we're going to say this is a mechanically gated channel. Make it a little easier for you guys. Mechanically gated channel. So we're forcing this gate open by pressure. Now we clarify that it's a sodium channel. What do we know about sodium? Well, we need to know a couple of things. We know that in all cells, outside of the cell is bathed in this fluid environment, specifically from your blood. So all of your cells are bathed with blood or cerebrospinal fluid or some sort of fluid. And 99.99% of the time, there's going to be relatively high concentrations of sodium outside of the cell compared to the inside, which will be very low in sodium inside of the cell. Now I'm going to continue on and talk about a couple other ions. As another example, we know that there is very high potassium inside of the cell and very low comparatively outside of the cell. And then finally, the last one we'll talk about is going to be calcium. We know that calcium is very high outside of the cell and very low inside of the cell. Why am I telling you these things? Well, because whenever we open a gate for one of these ions, they are going to flow high to low, always. That's diffusion. So in this case, we're opening up a sodium channel. Sodium is high outside the cell, low inside. Sodium is going to flow into the cell. So I'm going to draw sodium coming into the cell. Does that make sense? Okay. As an example, what if we opened up a potassium channel? Well, in that case, potassium would flow out. What about a calcium channel? Well, in that case, the calcium would flow in. Why can't they flow in already? Well, because we have a membrane that does not allow ionized, charged things in and out, because this is a very non-polar membrane. Okay, so if you need more clarification on that, you can watch my cell biology video here. It's really helpful. Okay. Now moving on, we've got sodium coming into the cell. Let me show what that would do to your neuron's voltage. We remember the neuron was starting out at -70, resting potential. But at this point, we have a stimulus opening up sodium channels. Sodium flows in. That will make the cell a little more what? A little more positive, right? Because we're gaining a positively charged ion. So I'm going to write this one as maybe number 1. And we're going to correlate that to the membrane voltage of the neuron at number 1, where we're starting to get positive because of the influx of sodium. Now, this is where things get really interesting. This is the stimulus, but again, we're not positive enough to do anything. In fact, at this point, we're somewhere around negative, maybe 55 millivolts or just under. So nothing's really happened yet because we wanted the neuron to get positive. We're still relatively negative. So we need some extra help. Check this out. Once this neuron, via those sodium channels, partially bringing some in sodium, we're going to hit something called a threshold potential. That is at -55. So -55 millivolts is called the threshold potential of a neuron. Now, think about it. Threshold. You cross a threshold, it's in a way a point of no return. So maybe you've seen the like married couples, like the husband picks up the wife and carries her across the threshold, and it's kind of the point of no return. You're married, you're done, like you're doing it now, right? So in this case, same thing with the neuron. Once it hits this, there's no turning back. We're gonna do something crazy. This is what happens. We are going to open up something called voltage-gated sodium channels. So number 2, we're going to have voltage-gated sodium channels open. And you have a ton of these. You have a ton of voltage-gated sodium channels open, and they're going to open at about -55 millivolts. So if we fly these guys open, you can predict what will happen. If these guys fly open, what's going to flow? Well, sodium's going to flow, and it's going to flow very rapidly into the neuron. By doing that, we will make the neuron a very, very, very positive charge all around, okay? So we're going to draw positive charges all the way around here. Okay, so what will that make the graph look like? Well, we know we're getting really positive now here at number 2. And that's because those voltage-gated sodium channels are opening. So we fly upwards and we hit a point called the action potential somewhere around positive 10 millivolts. Again, that is called an action potential because an action potential is a flow of positive charges, right? Now, here's the thing. Let's stick on the action potential just for a brief moment. Look at where I drew the positive charges. I drew the positive charges accumulating in the cell body. This is called the soma, okay, the cell body, the main part of the cell. But we need the positive charges to flow where? Down an axon, down this part, so that we can hopefully release these neurotransmitters. So that is exactly what will happen. All these positive charges will funnel down into the axon. Now, depending on if it's white matter or if it's gray matter, if it's white matter, we would have this myelin here. Remember the myelin, the insulation wrapping, so that the action potential can travel faster. So if it's in white matter, we're going to have myelin here, and it's going to make the signal travel really, really fast all the way to the end of the axon. Okay, very important. Now, at this point, what's the goal? The goal is to push these vesicles out containing the neurotransmitter. We need an ion that really likes to push things, and that ion is calcium. So when action potential reaches the end, we are going to open up what we call voltage-gated calcium channels. Voltage-gated calcium channels. I call all of these the Harry Potter channels because it looks like the forehead scar, right? So anyway, besides the point, voltage-gated calcium channels open at the action potential. Okay? So what will happen? These guys open up. Okay, if they open up, where will calcium flow? We talked about this. Calcium is going to flow in. Now I want you to remember, calcium likes to move things. Calcium likes to move things. You see this in the muscular system. Calcium moves the troponin and the tropomyosin gets pulled off. Calcium likes to move things. In this case, calcium likes to push synaptic vesicles out. The synaptic, if I can speak, the vesicles will fuse with the membrane because these are just membrane-bound organelles. It will spew out all of the neurotransmitter into this space. Neurotransmitter will bind to receptors on the target cell, and the target cell will have some sort of response. Once again, that response could be stimulatory, if it's a stimulatory neurotransmitter, or inhibitory, if that's an inhibitory neurotransmitter. Regardless, there will only be one neurotransmitter per neuron. So as an example, maybe this is a neuron that releases serotonin. In that case, this neurotransmitter would only be serotonin. There would be no acetylcholine, there would be no dopamine, there would be nothing else except for that one neurotransmitter. So there's only one neurotransmitter per neuron. That would probably be a test question if I were the teacher of your class, okay? So we did it, y'all. We got the neuron positive. We sent the action potential. We released the neurotransmitter. We communicated to the target cell, but there's a problem. Remember, it's just as important to turn something on as it is to turn it off. In fact, if this neuron were to gain a lot of calcium and gain a lot of sodium and stay positive for any period of time longer than this action potential, let's say it goes like this, the cell would actually die. If the cell stays positive for too long, it would die. So we have a problem on our hands. We sent the signal, but this cell is at risk of dying for being positive for too long. So we need to get this cell negative just as fast as we got it positive. How do we do that? Well, we need to make the neuron negative again. How do we do that? Well, check out this guy. First off, when we open these at negative 55 millivolts, we will also close them at the action potential, so at positive 10 millivolts. These calcium channels will close once we get negative, and we're going to see what happens here in a second. So we close these guys up. So these are back closed. I'm just going to draw them sealed off. So we're not gaining any sodium, but we're still positive. So interestingly, at positive 10 millivolts, these guys will open. These guys are called voltage-gated potassium channels. And these will open right up. Now think about it. If we open up voltage-gated potassium channels, where will potassium flow? High, inside the cell, out of the cell. So potassium is going to flow out rapidly. You have a ton of these. You have a ton of these voltage-gated potassium channels. So now, think about it. We're not gaining any more sodium, and we're losing potassium. What will that cause? that will cause what's called repolarization, okay? And this is going to be number 3. Voltage-gated potassium channels open up at that positive 10. So again, just a couple terms you may get tested on. This is called repolarization. We're making it negative again. And the other side on number 2, I didn't write this, but this is called depolarization. Depolarization, where we're getting very positive. So we're repolarizing by losing potassium. All right. Now, what's interesting is you see how potassium makes it very, very negative, and they will actually close at resting. I'm just going to say close at resting potential, which will be somewhere around negative 70 millivolts. So now these guys close up. So at the end of all of this, calcium will also kind of close up. These will relax. Don't worry about the end. They will reset themselves. So we're not going to focus on here. We're going to focus on the cell body. We close these guys up. We close these guys up. Neuron is negative again, so I'll redraw all of these negatives. But there's a problem, and I want you to see if you can find the problem. The problem is, even though this whole neuron is now negative, what did we gain a lot of, and what did we lose a lot of? Think about it. We just lost a lot of potassium. We just gained a lot of sodium. That's a problem because we needed sodium high outside the cell to flow in. We needed high potassium inside the cell to flow out. So we essentially need to reset this whole process. How do we reset it? I'm glad you asked. We have a very, very important protein called the sodium potassium pump. Sodium potassium, specifically ATPase pump. We'll write all that in. What's this guy? Well, anytime you see ATPase, it means on the protein itself, there is an enzyme that will clip off the phosphate of an ATP molecule. That just shows that we're releasing some energy. We're going to do some sort of work for the cell anytime ATP goes into ADP and a phosphate. What that will do will pump sodium, which we gained, back out of the cell. Specifically, if you want to know, 3 sodium. And into the cell, we will, as you probably guessed, pump potassium back in. If you want to know, 2 potassium. So by pumping out sodium, pumping back in potassium, we reorient the membranes, basically concentration gradients of sodium and potassium. That will be happening, we'll say at number 4. The sodium potassium pump fires and reorients the membrane. Fascinating. Now here's the thing. With action potentials, they will always look like this, where we had slight depolarization, rapid depolarization at threshold potential, and then rapid repolarization, sodium potassium pump will fire. Now, that being said, we've just reset the neuron, okay? So after we reset it, this is called the refractory period, basically where we can't send another action potential. But hypothetically, what if we get another stimulus? We could send the same action potential and do the same thing and send another action potential. So depending on how strong the stimulus is, we will have what's called frequency modulated action potentials. Let me just draw it right here because I've got some space. Let's say there is a very, very, very strong stimulus on this neuron, like a spider bit you and it's really painful and they're sending a lot of signals. In this case, your graphs of action potentials would look like this. It would just send so many action potentials per second. Whereas if it was just kind of a light brushing, like a very light touch on your skin, it might look a little more like this, where you just have one action potential and then another action potential. This is what happens when the neuron is very stimulated. This is what happens when the neuron is not very stimulated, right? So that's called frequency modulated action potentials. So we know neurons release neurotransmitters when stimulated, but how can neurotransmitters maybe oppose each other? How can we have maybe a stimulatory and inhibitory one communicate to a neuron? What would the neuron do in that case? We need to figure that out first, and then we're going to talk about how the brain and spinal cord actually integrate all of this stuff together on a very practical level. All right, so now we're going to go into how a couple of different neurons can affect a single one, talking about graded potentials, and then we're going to kind of connect it to a good clinical example in the spinal cord as well as the brain, and then we're going to jump into brain anatomy and physiology. So first off, we've got these neurons here communicating to this black neuron here, and we are showing the charge of this black neuron on the graph. So right now we're going to say this neuron is at resting potential -70. Now, we consider this neuron the postsynaptic neuron because it is coming after a synapse, which is the space where we're communicating from one neuron to another cell. So here's the synapse, postsynaptic neuron. That would make these two neurons presynaptic neurons. So both of these guys are going to be presynaptic neurons. Now, this is really common in the central nervous system because this is where we're communicating between interneurons, so one neuron to another, and we can influence that neuron in a couple different ways. So as an example, let's say this blue neuron is going to fire. It's going to send that action potential, and it's going to release a neurotransmitter. Now, let's say that this is going to be an inhibitory neurotransmitter. In this case, if this is acting as an inhibitory neurotransmitter, we are going to oftentimes bind to these ligand-gated potassium channels. Okay? Ligand-gated potassium channels. What that means is when the inhibitory neurotransmitter binds to the receptors, this gate will open and it will allow a passageway for potassium. Now, if we remember from the previous example, potassium is always high inside the cell, so it's actually going to flow out of the cell ever so slightly. So if we are losing a positive charge, what will happen to the neuron? Well, obviously in this case, it's going to get more negative because we're losing potassium. Okay, so we call this going down hyperpolarization. And your instructors may call it an inhibitory postsynaptic potential, because we're again talking about the postsynaptic neuron and what its voltage is doing in the membrane. Now, we consider this anytime we're just kind of adjusting the resting potential of it below that threshold potential. These are going to be called graded potentials. We're kind of grading them up and down, but we're not actually sending the action potential. Now, this makes sense because if we're down to say like -80 or -90 now, this neuron, this black one, is less likely to fire, less likely to get positive and send its signal, right? So that's why this is an inhibitory neurotransmitter. Now, let's say instead, now we have this guy, he's going to send his action potential. We are going to release a neurotransmitter here, but in this case, you can probably guess, we're going to consider this guy a stimulatory neurotransmitter. Now, you guys are smart. In this case, we're likely going to open channels dealing with sodium. So now we open sodium channels. We know sodium is high outside the cell. It's going to flow into the cell and make it maybe slightly more positive. So in this case, we call this slight depolarization. So we're getting positive. So we call this an excitatory excitatory postsynaptic potential, where we're going slightly upwards. But again, we're not reaching action potential yet. So we can see how presynaptic neurons can influence the postsynaptic neurons' voltage. And obviously, if this guy continues and continues, we're going to reach that threshold potential, hit action potential, and then send that action potential before going back down, just as always. Now, how does this actually work, though, in your body? Well, let's give an example of this. Let's say you are an athlete and you have some muscle tissue out here, and you had that muscle tissue damaged. So maybe you worked out really hard the days before, and maybe some of these fibers are kind of torn up and just basically ripped apart, because that's what you do when you work out a lot. And that's a good thing because we'll build them back stronger, but we know that we will have neurons that will detect that stimulus. So we're going to have a neuron out here. This is going to be the dendrite of a sensory neuron. It's going to come in to the spinal cord and synapse with another neuron. Now, again, this is a sensory neuron. We're sending action potentials towards the central nervous system about some pain in a muscle of our body. We enter that spinal cord and we're going to have another neuron. Oops, I'm not going to draw it in red. We're going to have another neuron that will send its action potential upwards, up ascending tracts of the spinal cord, synapse in the thalamus, where the thalamus will then send it up to the parietal lobe specifically, and that's where we're going to process, again, up in the gray matter of the cortex of the brain. Now, in this case, think about the types of signals we're sending. In this case, we're probably going to stimulate this neuron, stimulate that neuron, and then stimulate those neurons up here. So all of these neurotransmitters will be positive or stimulatory neurotransmitters, most likely, right? Because we're sending that signal. Now, what signal are we sending? Well, we can think of this signal in a way as being perceived as pain, right? Why? Well, we have damage, so we interpret that as pain. So what we call this is a sensation, which is the stimulus on the receptor, and then we have the active conscious perception happening in the gray matter of the brain when we perceive what the heck that stimulus is. Now, what's really interesting is occasionally what we can do is exercise again. And have you ever had this happen before where you have like a sore muscle, you go and work out, and then all of a sudden the pain kind of disappears, like you don't feel that soreness anymore? Well, what's going on there? Your muscle is still torn, but you can't feel the pain as much. What's happening here? Well, in this case, we sometimes can activate descending pain pathways. So I'm going to draw a descending pain pathway coming all the way down and communicating to this guy right here, okay? So again, this is called the descending pain pathway. You may talk about it in class, pain pathway. And this neuron system sends down something called the periaqueductal gray matter, and then we send it all the way down to this interneuron on the base. Now, in this case, when this system gets activated, especially during like exercise, during meditation sometimes, these things, meditation, these descending pain pathways can become stimulated. So what happens here? This blue neuron stimulates this guy to stimulate this guy. But in this case, when this blue neuron sends its action potential, it's actually going to send an inhibitory neurotransmitter. What will that do to this, in a way, postsynaptic neuron? Well, it will make this guy less positive. It'll make it more negative. It'll hyperpolarize it. What does that mean? Well, this blue neuron then is inhibiting this neuron. So if we inhibit this neuron, we're not sending that action potential as often. And if we're not sending that action potential as often, we're not basically perceiving that pain anymore. So this is something called sensation versus perception, and it can be altered, the perception, depending on other presynaptic neurons influencing the neuron that's actually sending that signal in the first place. So again, this is how we can kind of interact one neuron to another. Now, one cool thing you've probably heard of before, these inhibitory neurotransmitters are actually called endorphins. So if you've ever had that, what you call endorphin release or endorphin rush, that's basically when you're not feeling as much pain because the descending pain pathways were activated and you're inhibiting all these pain signals going up to your brain. Pretty fascinating. Okay, before I send you to the brain structures and functions, let's go through 3 questions that you may get on a test. So the first one, how many neurons are involved in a typical reflex and where do they travel? So we know reflexes are very similar to the following analogy. We hop onto an elevator and then we immediately turn back out of the elevator, okay? Well, what is the elevator in this case? Well, it's always going to be the spinal cord. So in the spinal cord, we know that we're going to be detecting stimuli, and that stimuli is going to be sent into the gray matter of the spinal cord. This is, again, the sensory information. So as an example, let's say this is like your patellar knee reflex. So let's say this is like your patellar tendon, and we're having some sort of force on it, like a hammer when you go to the doctor, and we have a sensation going in to the spinal cord. Now, typically, we've talked about how that neuron will communicate synapse with another neuron, which will travel up and communicate again to the outermost portion of the brain eventually to process it. But in this case, we actually decide, hey, we need to send a reflex outward to contract the muscle so that we do not damage that tendon, because if this tendon gets stretched too much by that force, it will actually tear. So we need to contract the muscle back so that it prevents that tearing. So what will happen is this neuron, sometimes this neuron itself that we communicate to has another axon. And that other axon can directly synapse with a motor neuron. And that motor neuron will come out and communicate directly to that skeletal muscle to contract it. So in this case, we have sensory neuron, interneuron, motor neuron. So the typical reflex includes 3 neurons. And where do they travel? We know that the sensory neuron will travel through something called the dorsal root of the spinal cord. And the motor neuron is going to travel through something called the ventral root of the spinal cord. So if we were to cross-section the spinal cord and look at that, this would be the dorsal root coming into the spinal cord. And then we'll have the ventral root coming out of the spinal cord like this. So again, here's the spinal cord right here, spinal cord. Here's the dorsal root. This is where that sensory neuron axon is going to be coming in, communicating into the gray matter of the spinal cord. That's where that interneuron is going to be. That interneuron is going to directly synapse with that motor neuron that will then travel out through the ventral root. So once again, this would be like the dorsal side of your spinal cord. This would be the ventral side of your spinal cord. Very cool. Now the second thing, how do spinal cord injuries affect the peripheral nervous system? Well, peripheral nervous system is all about sensation and motor output. So we know if we damage the spinal cord at any level, let's say right here, we are going to cut off any ascending, right, sensory information neurons. So therefore, we can't even get the information up to the brain. So we won't be able to feel any tracks that are basically traveling below that damage. So in this case, if it's really high, maybe like a cervical 5 vertebrae, we probably won't be able to feel from about the shoulders down, which is pretty nasty. Not only that, but we know the motor output has to come from the brain down and then communicate with the motor neurons. But in this case, again, they're severed. So both sensory and motor divisions will be damaged depending on where that damage occurs, and anything below that you will not be able to feel or move, which is really, really nasty. Now the last one, what regions of the brain deal with the perception of the senses? I'm going to send you over to the cerebral cortex video. It's going to go over the brain, but if you're interested, you can also check out my spinal cord videos, my cranial nerve videos, or sensation versus perception, or any other videos about the nervous system that I have on this channel. So best of luck, keep working hard, and well done.